The world ain't all sunshine and rainbows.
So welcome everyone. Welcome everyone to this year's Demo Day, which is our second Demo Day. We had one in November last year. As you probably realized from this kickoff movie here, BII is about bringing innovations forward and for the benefit of society. And we have several different programs. One of them is the Business Acceleration Academy, of which we are going to see the culmination and the graduation of 4 teams that have gone through 11 very hard weeks. During these 11 weeks, they have been trained to sharpen their business plan in terms of IP, in terms of management, market analysis, and so on. And what you will hear today is 4 fantastic pitches on their stories that they are, of course, eager to bring forward. At last year's Demo Day, we awarded the BII Acceleration Award to Circle Biomedical for their progress during that 11 weeks that they went through. This company has actually progressed further and entered also our creation house and received a convertible loan and is now operating in the BII. Today we will pass on the trophy to one of the BAA team from this batch. So to kick off our pitches, I would therefore like to welcome last year's winner, Fredrik Pettersson Madsen, CEO of Circle Biomedical, to the stage, please. And it's not because I won, but it's because to illustrate, we will now put it over here, so it's ready for this year's winner.
Thank you. All right, thank you for the opportunity to present. My name is Frederick Madsen. I'm the CEO of Circle Biomedical, a mucus engineering company developing a novel non-hormonal contraceptive for women. Today, 125 million women in Europe and the US use a contraceptive. The last groundbreaking innovation in contraception was in 1960 with the introduction of hormonal birth control. It has gone on to revolutionize society, and it's by far the most used technology today, but it's also an old and crude technology. Today, more than 28 million women in Europe and the US say that it's extremely important that their contraceptive doesn't contain hormones. The driver of this trend is increasing awareness of the side effects. And interestingly, also, we're getting supporting evidence from the individual stories in science. Oh, does it change? Yeah. A recent study, including data from 1 million women, show that the risk of developing a depression is increased by up to 80%. The problem is that today there's a lack of alternatives in the market. The products that are convenient to use are highly invasive, by either being a systemic drug, an implant that causes inflammation. On the other hand, the products that are not invasive are inconvenient to use. Our mission is to create a new technology addressing this gap based on the idea of mucus engineering. Cervical mucus is actually impenetratable to sperm cells throughout most of the menstrual cycle. It's only on and around ovulation that hormonal changes cause the mucus to loosen up, become more watery for sperm cells to penetrate. We've developed a technology that cross-links the ovulatory cervical mucus to reinforce this natural barrier and prevent sperm cells to enter. We have proof of concept on this idea. In the video you see our very first proof of concept using model mucus, where we could clearly see there is an effect. This motivated us to pursue the idea. We set up a collaboration with the Karolinska Institute in Stockholm, where we started acquiring human ovulatory cervical mucus. We created an assay mimicking the cervix, where we looked at sperm penetration. And as you can see, with the treated mucus that have been exposed to our formulation, we're achieving 100% blockage. This effect is also almost immediate. And this is the foundation for our first product concept called We. With this first product concept, we are targeting a vaginal capsule that after insertion rapidly dissolves, releases the formulation, and we're targeting that the product works after 1 minute and for up to 24 hours. This is a groundbreaking product because first, it's minimally invasive. We only modify the naturally occurring mucus layer. Second, it provides complete freedom to the user. You can use it in the moment, and it provides extended protection. And finally, we're designing it with the user to create a great experience. Because contraception is about two things. It's about efficacy, but also about the user experience, which leads to compliance. Our product is classified as a medical device in Europe, and we also anticipate classification as a medical device in the US. The FDA has also released a guideline for vaginal barrier contraceptives, which defines the clinical path to market, which includes 2 clinical trials and a total of 250 subjects. We are targeting women that do not want to use hormonal contraception and prefer an on-demand method with this first product. If we combine the market potential in Europe and in the US, we arrive at a very sizable opportunity. We also want to disrupt the way that we bring the product to the market. So we see 3 exciting ways of bringing this product to the market. There is a traditional partnering with the companies that are already dominating the industry, but there's also a number of new online platforms that provide online access to contraception that we could potentially partner with. They have millions of users on their platform. They're rapidly growing, and we'll be able to access these. And you can also imagine creating a direct-to-consumer product and brand, leveraging this same online go-to-market strategy. We are focusing on building a team of A players from the medtech industry and from academia. As mentioned, my name is Frederick Madsen. I've been working in the contraceptive space for the past 5 years, developing an in-depth understanding of the user needs and the commercialization process. Thomas Croucher is my co-founder. He's a scientific co-founder and CSO. He received his PhD in France, and then moved to MIT, where he started working on mucus, seeing it as a material that we can modify at a molecular level, and amongst other, reinforces barrier function. We're also happy to announce Ludmila Kachan to the team. She comes from UC Berkeley, and also very proud to announce Lisa Rarick as an advisor. Lisa is an OB-GYN by training. She worked for 15 years at the FDA, where she was division director for the Women's Health Office. And just to wrap up, a few words about BII, because BI has had a major impact on our journey. So we started getting into the BAA program, where we matured our business case. We're able to close an investment with a US fund. We then progressed into the Creation House. So now we have a real company, and we are transforming science into a product that can have a real impact in the world. Thank you.
So thank you very much, Frederick, for this quick update. It's a true pleasure for us to see how our graduates develop, and we are happy to be part of this journey. That must be difficult to say goodbye to this wonderful trophy, but today it's our job to put it again in other good hands. So stay alert. Now I will take you quickly through the agenda for the day. We will start with the BAA graduate competition. You will see presentations from 4 teams, and afterwards we will ask each and every one of you for a vote. And I will go into more details about that later on. Afterwards, while we will be busy counting the votes, you will be entertained by our current 2 incubation teams. That's in Proter, who you also might know from the previous Business Acceleration Academy program, and Caria Pharmaceutical. Afterwards, we will announce the winners, and we will go for a small reception at Bruce, just behind the corner of Empire Bio. So this is what's gonna happen today, and now let's move on to the BA presentation part, the major reason for us being here today. In order to get a better understanding of what the startups do, we will start with a short intro movie about each team, and then they will make a 7-minute presentation about their business and the status, and then we will have a panel that will help you make up your mind. So our fantastic panel consists of Lene Gerlach, who is currently building an early-stage engagement for life science startup at Växtfonden. We also have Roger Franklin, who arrived to us from Stockholm. He's a partner at Hidden Ventures, investing in the Nordics, but also moving on to Europe currently. And Patrick Dirks, Vice President from Christian Hansen, with more than 22 years of experience in industrial biotech. They will give a little bit of commentary, question, or suggestion to every single team that will present. But please remember, it's up to you to choose the winners today, so listen carefully. And let's start with the presentations. Thank you.
Hello, my name is Wilhelm. I am the CEO of Bifrost Biolabs. At Bifrost Biolabs, we focus on making biology easy. So, I'm a biologist by training. Wait 1 second. I think the battery just... Oh! Maybe I'm pointing at the wrong place. Can you shift the slides for me? Okay, Next one. So, as a biologist, I've used a lot of time in the lab, and it's always frustrated me. I will have a solid plan for what I wanted to do when I went into the lab. Then I'll go into the lab, and I'll get stuck in trying to get the results I wanted, getting inconclusive results, and trying again and again and again to get some concrete data. Later, I started working with laboratory automation in the hope that that would be able to solve the problems with unstandardized work and long hours that I had. But I found out that my time was just shifted from being in the lab to now sitting in front of a computer screen trying to code these complicated machines that I was using. So I thought, isn't there some way where you can make simple machines that everyone in a lab can use that can solve the most time-consuming tasks in the lab, such as cell growth? That's exactly what we did. This is our first product, a small sensor that you can put inside any transparent vessel, such as a shake flask. It can give you real-time online data, so you always have access to your data. And this is how the system looks, because we're not only creating one sensor, we're creating a sensor system consisting of 16 sensors that are all controlled by our control unit. It takes 2 minutes to set up the system. It can run fully automated while when it's set up. It can run more shake flasks than any of our competitors, and all of the data will be accessed from your computer on your workbench, the smartphone in your pocket, or the tablet close to the incubator that you're working at. So, who is interested in this, and what do they actually want to use it for? Well, if our customers are interested in using this for scale up. So when you scale up a production, you start in a microtiter plate, you just made your new invention, and now you want to scale it up. Let's say you're Novo Nordisk, you just created a new type of insulin. Now you want to take it up into big steel reactors so you can actually earn money on it. This process can take up to 2 years and is very labor intensive. What our system does makes it possible for you to understand what's going on in the small scale. This is very important to be able to understand which parameters you need to know in the large scale, making it possible for you to understand the small scale, the black box of small scale, which is what our product does. It makes the black box into a transparent box of known parameters, making it possible for you to faster extract and understand the parameters so that you can shrink the time it takes for you to scale up. So we estimate from talking to industry partners that every department that implements our technology will be able to save up to 2 million kroner. Novo Nordisk has 15 departments here in Denmark. So who are these potential customers that I'm talking about? We have companies, or we have people from academia and people from the industry. In total, we have 7 letters of intent. The general market is quite conservatively estimated to be here in Europe, 2,300 potential customers, similar number in the US. The patent. I'm very happy to announce that we've just delivered our patent, and we own all of the rights to our IP. So, it's not only me making this possible. We have a very strong team with a wide variety of competences, such as Hasse, who has 15 years of experience within sales and business development within laboratory automation. We have Jesper and Paul, who have a lot of experience in creating sensors, primarily for the mining industry, but they're able to translate that knowledge to create our product. Then we have Suresh, who has 10 years of experience in bioprocess monitoring, so he understands what our customers want and how we're able to deliver that to them. Then we have Lars Ulrik, who has 15 years of experience in creating products for creating products using photonic measurements, as we do. And we have Jan, who has over 30 years of experience in the life science sector and who understands how we are supposed to plan and reach our milestones in the future. So how does the future look? Well, currently we are delivering our first prototype to Novo Nordisk here in August, and we expect our first product to be ready for sales in December. And we are now looking for funding to make that possible, to breach the last gap here. We're looking for 3 million Danish kroner. We have the team, we have the patent, we have the product. Now we're just looking for the investment to make the last part possible. If you're interested in helping us, then please contact me on my email address or come talk to me afterwards. We have a small demo with us so you can actually see and touch our product. Thank you very much.
Thank you very much for the amazing presentation. And do we have any comments or questions from our panel?
Thank you. Can you hear me? Thank you, Wilhelm, for a very nice presentation. And we are already in dialogue. And I can see that we are on your slides. So that's nice. Thank you for that commercial. I was thinking about the differentiators compared to your competitors. Can you elaborate a little bit on those? You said more parameters you can measure, you have more sensors, but can you elaborate a little bit on your differentiators compared to your competitors?
One of our key advantages is the flexibility. So our system can be used in any transparent vessel. Of course, now we're looking at a shake flask, but it also means that you can use it in a falcon tube up to a 5-liter fermenter. So fermenters, falcon tubes, or shake flasks. This is flexibility that none of our competitors have. So.
Great. I was wondering whether you could provide a bit more color on what the nature of the parameters are that you're monitoring. And how much robustness that provides you with in terms of the, how many scale-up failures do you experience with your technology versus not using your technology?
Well, I can't say for sure because our clients are not testing our product right now, so I can't know exactly. But what I know is that one of the key players in the scale-up market says that the industrial failure rate is 10%. So and they say that it takes 2 years to scale up, and that time can be shrinked if you have a better understanding. And with our technology, you can run 3 times as many screenings in parallel as you would be able to do manually. So that means that in principle you'll be able to increase the speed 3-fold. Yes.
Thank you, Wilhelm, for a very nice presentation, very energetic and very person-driven by your own challenges, so that's now very nice to see. As a company, as representing a company living on fermentation of microbes, measuring things like OD, cell density is of course key to our business. So I think it's very nice to see an alternative technology popping up in this space. A bit of a question. You mentioned you had 16 sensors. So one of them would measure optical density. The other ones, what would they do?
Oh, they all measure optical density right now. In the future, we're gonna be like increasing the parameters, but currently it can only measure this one parameter.
And it's, if I understood correctly, it's a sensor which is glued to the inside of the tube or the vessel?
Well, as you can see the picture here, it's inside the vessel. It's not glued. You just put it in, and that's pretty much it.
So it's floating around in the fermenter?
Well, then we would get into the nitty-gritty, which you are very welcome to come talk to me afterwards. Then I can show you the demo. It will all make much more sense.
Great. Thank you very much.
Thank you, Wilhelm. Now we can move on to the next team, Proxy Biotech.
Hello everybody, I'm Kasper, I'm CEO of Proxy Biotech. First of all, thank you to the entire BI team for allowing us to pitch here today. So, at Proxy Biotech, we have developed a detoxification platform for detoxification of bacterial toxins. We call this the BID technology. We can take any bacterial toxin, detoxify it by use of our BID technology, and then we have a toxoid. This toxoid can be used as an antigen in a vaccine. And once you inject it with the vaccine, you are protected from the disease symptoms caused by the toxin. Now, toxoid-based vaccines is not a new invention. In fact, the idea of using toxoid-based vaccines has existed for almost 100 years. And the way we develop many of our vaccines today are still by the use of this ancient method, a method that's based on formaldehyde. And when you use formaldehyde, you create some major challenges, but some challenges that can't be avoided by the use of Proxy Biotech's BID technology. So, first of all, formaldehyde is a carcinogen. With the use of BID technology, we only use non-hazardous reagents. Second, formaldehyde detoxification is a reversible process, meaning that the toxoid can actually revert back to the toxic state. Using BID technology is completely permanent. It cannot revert back. And third, and most importantly, when you use formaldehyde, it results in a lot of modifications on the surface of the proteins, meaning that the immunogenicity you get is low, it's not optimal. And due to this low immunogenicity, formaldehyde cannot be used to detoxify a range of bacterial toxins, including those causing whooping cough, botulism, and Clostridium difficile infection. Using BID technology, on the other hand, results in only very few modifications of the surface protein, leading to a high optimal immune response. So, to validate our technology, we have developed a vaccine candidate targeting Clostridium difficile infection, CDI. CDI is one of the most common hospital-associated infections and has a mortality rate above 8%. Furthermore, the economic burden is tremendous. Every year, we spend more than $10 billion just to treat the symptoms caused by CDI. And as you can see in the graph in the middle, this is not just a problem today. This is a problem that's going to increase in the future. Simultaneously, we also see a almost threefold increase in the market value, reaching more than $1.6 billion in 2026. Vaccines are going to account for a third of that market. So the vaccine candidate that Proxy Biotech has developed shows some very promising results. And to support my statement here, I brought you some data. Now, we're not digging into all the details here, but what it shows is that our toxoids get a very strong antibody recognition. We get a high immune response when tested in animal studies. They're very preserved, and they're safe. We see absolutely no toxicity left in the toxoids. And these are the 4 essential properties that a toxoid needs to possess in order to be used as a vaccine antigen. We also know that there is a huge demand from the industry to get access to new technology like this. And we know it because 2 months ago, we went to the World Vaccine Congress in Washington, D.C., where we met with decision makers from some of the biggest vaccine companies in the world, including Pfizer, Merck, and Sanofi Pasteur. Now, Pfizer, they are leading with a phase 3 vaccine candidate targeting Clostridium difficile infection, CDI. And the technology they use today to produce their vaccines is very similar to the formaldehyde approach I just mentioned before. And that really complicates the manufacturing process. And they've expressed interest in implementing our BID technology in-house for production of their CID vaccines in the future. Merck, they have also expressed interest in implementing our BID technology in-house for the development of AMR-targeted vaccines. And finally, Sanofi Pasteur. They're also very interested in to get access to our technology for the production of various vaccines, in particular for the whooping cough vaccines. So, to validate our technology, we have designed an activity plan in alignment with the requests from the industry. In addition to our CDI vaccine candidate, we're going to apply our technology on the toxins causing diphtheria and whooping cough. And the reason for choosing these toxins is not to make a vaccine targeting the disease for the market. It rather serves 2 purposes. So first of all, it allows us to run quick and dirty studies in head-to-head with commercially available vaccines. And second, The toxins causing CDI, diphtheria, and whooping cough, they are all very different when looking at their composition and their size. If we can demonstrate applicability of our technology on all these toxins, meaning we can detoxify them and raise a high immune response in animal studies, we actually demonstrate that our technology can be applied to a very broad range of bacterial toxins. And reaching this point, our proof of concept inflection point in early 2020 allows us to engage with the industry. Following this, we are going to identify new vaccine candidates to develop. Our focus at the moment is on maturing the detoxification technology to detoxify bacterial toxins. However, there's absolutely no reason not to believe that this technology cannot be used in other areas, such as production of vaccines targeting viruses or venom. Hence, the technology has a very big potential. A big potential that's created by a very competent team. In addition to myself, we have René and we have Aria. René, he's a senior scientist at Statens Serum Institute, and he's also main inventor of the technology we use. He's an expert in bacterial toxin and has extensive experience in both supervision and planning of research activities. And then we have Aria. Aria is a PhD student at Statens Serum Institute and co-inventor of the technology. With a background in biotech engineering, he has a profound experience in the detoxification of bacterial toxins. So right now, can you switch to me? Thank you. Okay, anyhow, we're looking for 3 million kroner that allows us to reach our proof of concept inflection point, bringing our technology to market. So if you find this interesting, or you think you're the person, or you know the person who can help us to bring this to the next level, please contact us after the presentations or on our email address here. Thank you very much.
Thank you very much, Kasper. That sounds extremely promising for me.
It is very promising.
Now let's ask our experts what they think about that.
Thank you very much for a nice presentation, and we are of course also in dialogue.
Obviously.
So can you elaborate a little bit on your patent situation?
We can do that. So we own all the rights for the invention, and we are going to file next week for patent. Yeah. So we have the final draft now. We're just revising it final time, and then we're going to file it.
Okay. And why do you win the battle compared to your competitors?
We win the battle because the big pharma companies, who are the only ones who bring vaccines to the market, they have asked for this technology, basically. So we've talked with them, and they want this new technology for their production. So that's why we win.
I'm interested in the C. diff program and the discussion with Sanofi. I think Sanofi had a phase 2 failure with a
phase 3 actually.
Phase 3 failure with their C. diff vaccine.
Yeah.
And the opportunity they want to pursue with you, I think you said, was whooping cough. I'm just wondering if they discussed, or you have an idea as to whether that program failed due to issues with the toxoid, and whether you could help with that.
Yeah, I know that. So we talked with multiple people from Pfizer and also from Sanofi. So Sanofi are not, what they said is they're not completely sure why they failed. But we can see when we align the technology that Pfizer used versus Sanofi, they use the formaldehyde approach completely. Pfizer, they modified that a bit, and they get a better immune response. So that's one thing. And there's also something with the testing in the phase 3 clinical trials, with like the results they get there. Yeah.
Thank you, Kasper, for a very nice and clear presentation. It's a bit out of my comfort zone, but one question anyway. You mentioned that the toxoids, they attack the actual toxic compounds, so they don't attack the actual bacteria. Is that correct?
Yeah. It is specifically for the C. diff. So Clostridium difficile express 2 toxins, toxin A and toxin B, and they give rise to the disease symptoms. So what you want, we know, like patients who have a high immune response against these 2 toxins, they're protected from the symptoms, while patients who has like a very low level, they get the symptoms. Yeah.
Thank you.
Thank you very much, Casper.
thank you.
And for coming. And let's welcome on stage third BAA graduate, Octarine Bio.
Hello everyone, my name is Netaji and we are Octarine. We are a synthetic biology company producing superior cannabinoids. We strongly believe these molecules have potential to improve health of millions, even billions of patients' lives. Cannabinoids are a diverse class of molecules that acts on cannabinoid receptors. There are more than 100 different cannabinoids isolated from cannabis plants and have shown health beneficial properties. Few examples are stimulation of appetite, reduce pain, and reduce anxiety. The market for cannabinoids are growing. The US pharmaceutical cannabinoid market is projected to reach $50 billion by 2030. This is a compound annual growth rate of 33%, driven by the positive legislation change in US and Canada. So, what is the problem with cannabinoid then? There are actually two sides to the problem. There is the suboptimal production and then the suboptimal products. The production of cannabinoid today is almost entirely driven by plant extraction. Plant extraction is resource intensive. Some of the important cannabinoids are present in minute levels. Then the quality varies each time, making it difficult to use it in pharmaceutical applications. Chemical synthesis is another option. However, the iteration from one to another cannabinoid is still slow and costly. Because of the chemical nature of these molecules, there are also drawbacks and limitations using cannabinoids in pharmaceutical industry. Some of the key challenges are lower bioavailability, insolubility, psychoactivity, and poor absorption. Octarine has developed enzymatic platforms to expand the chemical diversity of cannabinoids, allowing superior molecules. We combine this with the yeast engineering platform to scale up the production. Our combined technology would have a rapid generation of molecules, fast iteration from one to another cannabinoids. Our products are 99% pure, and both production process and superior cannabinoids are patentable. Our first enzymatic platform has more than 380 enzymes. This is a high throughput assay. We conduct small enzymatic modifications to the core structure of cannabinoid. Modified cannabinoids then would be subjected to an in vitro screening cascade where we select superior molecules with superior functions, such as more soluble, more bioavailable, non-psychoactive, etc. Now you may be thinking, how do they going to apply these molecules in the industry? CBD is today used for epilepsy treatment. There are two problems though. CBD has less than 10% bioavailability, and it is an insoluble molecule, making oral drug administration challenging. These are two problems that we can fix using our superior molecules. Cannabinoids are used for pain therapy today, and when you are in pain, you would like a fast response. When the cannabinoids are used in the plasters and patches today, the response is slow. This is because of the poor absorption. This is a problem that we can fix with our superior molecules. THC is a psychedelic molecule, but it has been used for more than 200 clinical trials today and show high potential to be a clinical and pharmaceutical drug. But psychedelic property is not desired by many pharma applications. Non-psychoactive THC is something that we can provide with our technology. If we look beyond the pharma, there is a huge interest for cannabinoids in consumer space, especially in the beverages. But none of us would like to have a Coca-Cola with oil taste or our beer with oil droplets. Insolubility is a problem that we can fix using our superior molecules. It is true there is fierce competition in the synthetic biology cannabinoid space. Luckily for us, our competitors are focusing improving the production of natural and rare cannabinoids. We are focusing on improving the products so the products are more efficient and accurate. The team behind all this is a group of highly skilled scientists from KU and DTU. The team has world-class expertise within yeast engineering, fermentation, scale-up, and as well as plant biochemistry. Both co-founders previously has run research projects all the way from idea to commercialization with industrial partners. Our advisory board, our excellent advisory board here, we have Morten from Pfizer representing the pharma industry. We have Ernesto from Firmenich representing the biotech industry. We have Biwa as a cornerstone in our science. We have oh no, okay. We have included KOLs to sanity check our ideas, especially within medical chemistry and CNS drug delivery. When we started with BIA, we pivoted. We had to shut down our lead projects. We ended up with the project that was far from proof of concept at that time. Now we have proof of concept achieved. We have IP filed, and our IP strategy is on place. We have interest from different funding agencies. What we look for is bridge funding until December, because our next goal is Creation House. To be specific, 2.5 million Danish kroner to cover our R&D and commercial development. We have interest from different pharma companies, and we have had face-to-face meeting with one of the biggest CNS pharma a week ago. We believe early stage collaboration with the big pharma is possible. Traction is important to us. It is a part of our business strategy. We are thrilled to be featured by SynBioBeta today in their newsletter, and we are also thrilled to be invited to the conference in San Francisco in October. With that, I would like to say thank you to BII and Novo Foundation for the amazing opportunity, and BII team and Christian for taking care of us, and for my amazing team for excellent work, and thank you so much.
Thank you, Netaji. That sounds like amazing market opportunity and very quick traction. And Patrick, it sounds like project more in your comfort zone, so we can start from you this time.
Thank you very much. I didn't know you knew that this was more closer to my area of expertise. I'm Dutch, sorry, so you know. That's why I have no user experience in this area. Thanks, first of all, for a very nice presentation and very clear too. There's actually two questions, because one question, it's a bit technical. We heard a couple of weeks ago in Zymergen's presentation at BII, 750 people doing synthetic biology, and you mentioned that they are not interested in this market share, or companies like Zymergen. If you could elaborate a bit on that, that would be one question. And to build on the second question, is consumers, they are more and more looking for natural products, non-synthetic, non-whatever. Have you thought about that trend in society?
Yes, I'll start with the first question about the competitors. We have conducted a comprehensive IP landscape analysis and as well as competitive landscape analysis. That's why we know that we are the first movers to improve the products. So far from the IP at least, we are well aware that our competitors are focusing on improving the production, higher titers of natural cannabinoids. When the second question, first of all, I would like to say our first focus is the pharmaceutical market, not the consumer market. I think it's in the consumer market where people are more focusing on natural products. Yep.
I have a question on, so what would the advantage of your approach versus the kind of traditional medicinal chemistry approaches in this area? So I'm not an expert in this particular area, but it'd be good to understand that. And secondly then, you know, you mentioned that your kind of in-cell enzyme platform expands kind of the chemical space. So I'd like to understand a bit more about how that happens, how that in-cell platform is set up.
Yeah. So the first question, compared to chemical synthesis with enzymatic way that we do, we have a fast iteration, meaning that we only need to add one more gene to get a huge variety of products. While in the chemical synthesis, you have to add more, and it's large scale. Changing from one molecule to another is much more difficult, which is not the case for us. Expanding the chemical diversity, we have chosen 5 different classes of enzyme families, knowing what they do, having experience working with them in other projects. The first class of enzymes is what I featured today and what we have patented, where we have 380 enzymes. This class of enzyme we have previously used, and we know with this modification we can achieve more bioavailability and more higher solubility. So that's why we are focusing on this class of enzymes for now. But the idea is to expand the platform. Yeah.
Yeah. Hi, and
Hi.
thank you for a nice presentation. As you mentioned, it's a very crowded area. There's a lot of players, large players, and you said that you have filed a patent application. But have you looked into the freedom to operate situation? There can be granted patents out there that actually dominates your product.
Actually, doing IP landscape analysis was one of the first thing we did at BIA, and it's part of our business strategy. We use the IP landscape analysis to discover our sweet spot. And this is also why we are not focusing on improving the production, but the products. And before we filed the priority application, we have conducted the FTO to see that what are the other patents out there. And we are convinced we have freedom to operate.
So a patent consultant has conducted
Yeah.
a freedom to operate analysis.
Yes.
Okay.
Thank you. And now let's welcome, last but not least, BAA team, Tres Action.
And that is really why Transactio is here. As you just heard, synthetic biology is amazing and it has great potential. And new technologies within genetic engineering are opening up new frontiers for a better and more sustainable future. But for any of this to matter, we need to be able to get these products out of the lab and into the world. When you want to make a new product, you typically start by generating thousands of candidate organisms. And it's then the job of scientists to take these and find that one organism that actually goes into production and has an impact. This is a really challenging problem. We went out and spoke with a lot of customers to hear how they do it today. And they do it in a funnel where they start with their candidates, and in a series of steps, they whittle them down until they find their next blockbuster. The problem lies in the middle of this funnel. Many companies still rely on shaker flasks. They don't provide any monitoring, and they can't control the process. So it's like looking for a needle in a haystack, blindfolded. The next step is lab-scale bioreactors. They do provide monitoring and control, but they're too slow and don't offer enough throughput to be used on their own. And this is where Transactio comes in. We want to fix this problem in upscaling. Our vision is to improve the cell culturing labs through automation and data-driven approaches, such that the scientists can do what they're great at, and machines can do the rest. We believe in having a big vision, but starting small. So we started out by talking to our potential customers, more than 10 in fact. And based on their input, we designed a minimum viable product. We spoke with Novozymes, who were particularly interested. And they saw the importance of what we're doing, so they signed a commercial letter of intent stating that they want to help us develop through pilot tests and of course purchase it once it's done. We've also spoken with Siemens Technology Accelerator, who have some interesting and unique sensor technology that they're willing to make available to us, because they recognize the importance and the value of the problem we're solving. So this came together to our design. It's completely wireless, sits on top of a flask. It has a sensor probe going into the culture, monitoring key parameters. And we have 3 liquid containers for controlled fed batch fermentation. But really central is, this is very easy and very fast to use. So the workflow is familiar to anyone that has worked in a fermentation lab. You start by autoclating the culture as well as our filter cap. Then you design your experiment using our easy-to-use software platform. Then you assemble it by clicking on the electronics and liquid containers. Lastly, you inoculate it, put it in an incubator, and you're good to go. And you can monitor everything online from the comfort of your computer. Comparing our product to what is available today, we see that in terms of features, such as the ability to control pH, add nutrients, and so on, we are very competitive with lab bioreactors. And of course, we blow shaker flasks out of the water. But when we talk with our customers, they say again and again, the things they really care about and what drives the economics is the data quality, of meaning how many strains can you really get good data on, the speed, so how quickly can you set up an experiment, and the cost, which essentially translates to how many experiments can you run in parallel. And here you can see our MVP already is a very attractive business case. There are other approaches to this problem as well, such as microwell bioreactors. They work with very small volumes, leading to several problems. For instance, you can't take as many samples as you might want during an experiment. There are parallel bioreactors, like the Amber 250 system, but these are very inflexible and cumbersome to work with, as well as prohibitively expensive. And lastly, you can outsource it through a contract research organization or service provider. This is also problematic because there's a loss of know-how and innovation by outsourcing these things. So in short, we reduce uncertainty when screening, which in turn provides improved production organisms for our customers, which improves their margins. We've did some market research and found more than 7,000 companies that conduct relevant kinds of research that could use our MVP. These companies already have a high R&D spending, and from talking with to our customers, we know that they're willing to pay to solve this problem. So that's what we're doing right now. Right now, we're focusing on bringing our MVP to market by partnering with large companies like Novozymes. This will allow us to mature our key technology. But really, we're interested in making a mass-produced product that we can get out there and collect a massive data set. And using this, we will pave the way for what we want to do next, which is a fully automated system that can set up and conduct an experiment without the need for human labor. Such a system will be able to fully take advantage of the new techniques within machine learning and data-driven approaches. This has an even greater commercial potential, and we believe this is what's needed to disrupt the strain development process and truly bring about the biological future. And to do so, we have an excellent team of diverse technical backgrounds, from data science, product development, sensor technology, electronics, and software. We're really covering all the bases needed to develop such a product. And to support our efforts, we have some very strong advisors with experts with expertise within commercialization and fermentation technology, as well as experienced entrepreneurs who've been through this before. So with their help and the help from the excellent BII team, we're right now validating the robustness of our technology and validating it in the market by talking with customers. Next, we'll integrate this into a limited feature prototype that we're testing at Novozymes. Once that's done, we'll develop the full MVP and test it more broadly with both small and large companies, universities and CROs, because we're really solving a general problem here. Once that's done, we're ready to set up production, which will take another 6 months. And then by the end of the third quarter, 2020, we can ship our first product. Up to this point, we've been able to rely on soft funding, and we've raised almost 1 million crowns. And we believe using soft funding, we can get all the way to the Novozymes pilot. After that, we need a new investment. Siemens Technology Accelerator, that I mentioned before, are interested in coming on board as an investor. But we would really like a local investor to come on board as well, that has some more experience with the industry and with the biotechnology side of things. At Transactio, we believe that just because biology is complex, the technology powering it doesn't need to be. So if you agree and want to help make this a reality, come grab me afterwards or shoot us an email, info@transactio.com. Thank you.
Thank you very much, Toke, for the fantastic presentation. And now, Roger, what do you think about the solving problem of strain selection?
Thanks for a great presentation. So I was just keen to hear more about the core technology here. So what are the parameters that are being measured? What's being manipulated? Can you go into any more detail about that core of how the technology actually works?
So the core technology revolves around some sensor technology that allows us to make very cheap single-use sensor probes. This enables a workflow that gives us this high throughput. We can monitor the pH, glucose, as well as the dissolved oxygen, which we have very good data on. And we're looking into some technologies for measuring the biomass as well.
And the IP around that is what? What's the, if you could describe the nature of the IP briefly, that would be great.
So there's some IP on the sensor technology, and we've done a freedom to operate analysis, an initial freedom to operate analysis with the help from SACO and through BII. That so we can see that we can do what we want to. There's also some IP that Siemens is interested in putting into the company that will give us further protection. But in terms of a defensible edge, a lot of it will come from the ecosystem and the data platform.
Great, thank you.
So on top of that you can follow the cell growth, you're getting data on the cell growth. What is the most important value proposition of your product? Is it time? Is it success rate of the cell growth, or what is it?
So there's a pressure on industry to have a consistent process where you can get it right the first time. So right now there's a lot of uncertainty in the process, and you risk either taking forward candidates that you shouldn't have, they need to go back, or maybe screening out the things that you were actually looking for. So you might actually end up dropping a blockbuster on the floor. I mean, on top of that, of course, we also help shorten the time to market by reducing the funnel, or making the funnel shorter. Does that answer your question?
So what is the most important milestone that you have achieved during this BAA program?
Well, we've done a lot during the BAA. I think it's really important for us that we know what we want to do as a company. When we came in, we had a problem and some technology, and really didn't know how to fit this together. So we have a lot better strategy right now, and we know that we're in a space that we're allowed to be from the freedom to operate.
Thank you, Toke, for a very nice presentation. When I went to university, I learned that you get what you screen for. So if you screen for growth in a shake flask and you optimize for growth in a shake flask, you get microbe which grows well in a shake flask. So have you any experience and any data on the fact that it actually scales to large scale production?
That's of course one of the key questions for us to answer, and we don't have that data yet. We've spoken with the labs in the Netherlands as well as in Sweden that can help us by providing access to pilot scale, because of course that's what it's all about.
And just one final question: is there a synergy with the Bifrost first presentation we heard?
There very well might be.
Thank you.
So now you have seen all 4 teams which today officially graduate from our first official BA program. Before it was a pilot, so today it's first officially approved by the board edition. And now you will have a task. We will ask you to help us choose the winner, and the winner meaning who has accelerated the most over those 11 weeks. And of course, all of you would have to ask many more questions to understand exactly what happened there. And you might probably be also affected by some sympathy to specific projects or people. And of course, it is subjective, and we cannot give you the answer. You basically have to vote for whatever you feel is right. In my opinion, all the teams are the winners, and all of them had their challenges, and they worked hard day and night on solving them. So all our babies are the winners today, but there is only one trophy, and only one team can go home with it. So please help us do the hard job, because we cannot. In order to do that, each of you got on your seat 4 bills. Each of them is worth 100,000 BII dollar, and you can distribute them the way you like. Outside this room, there are 4 boxes with names of the teams on it, and you can put all 4 bills into one box, or 1 bill in each box. This is your decision. We would ask representatives of the teams which are competing in this competition now not to vote. And we expect you to respect it. Now we will set up the timer for 15 minutes, and exactly in 15 minutes this door will close so that nobody will interrupt next presentations. So please make sure to be back on time. Thank you very much for your attention.
All right, all right, all right, all right. Wonderful presentations, huh? While we are counting all the BII dollars, we will have a few presentations now of some of our incubation companies in Creation House. The first one, he's known in the office as the Michael Jordan of biotech. It's Korea, who just joined the team, and the Creation House, we supply them with convertible loans offices and obviously lab space. But Ian, I think you're better telling the story. So please welcome Ian.
That's quite a new name. I haven't heard that before. Before I start, I'd very much like to thank the BII for scheduling this event on my birthday. And as my birthday gift, I understand I've been given a full hour to pitch to you today. Right? Relax, I'm only going to need 45 minutes. So my name is Ian Lakian, and I'm the CEO and co-founder of Korea Pharmaceuticals. And over the next 7 minutes, I'm going to try to give you just a glimpse of what we're all about. But in a nutshell, we're developing drugs for Parkinson's disease, which is the most common movement disorder in the world that affects over 10 million people. Take a moment to think about that number. Twice the population of Denmark. Parkinson's is characterized by a loss of dopamine-producing cells in an area of the brain responsible for movement and motor coordination. And as these cells die and the numbers decline, so too does our ability to do day-to-day things, like write, or walk, and eventually swallow. Currently, the way Parkinson's is treated is you address the symptomatic effects of lost dopamine simply by giving the patient more dopamine. While this is initially effective, what happens is the underlying cell death is still occurring, and as the patients decline, this treatment becomes less effective until it's not effective at all. Thus, an urgent and unmet need exists for us to develop disease-modifying therapies that help slow and hopefully one day stop the progression of this disease. For Parkinson's researchers like us, this is the holy grail, because nothing out there exists that does this. Or does it? This is the drug Byetta. That's the trade name. The molecular name is exenatide. And exenatide is a type 2 diabetes drug that helps diabetics control their blood glucose levels. It belongs to a family of drugs called incretin analogs and specifically acts on the GLP-1 receptor. Because we're a Novo country, I'd be remiss if I didn't show you another drug that you all know of, Victoza or liraglutide. Same mechanism of action. So why am I showing you these two type 2 diabetes drugs? And what does type 2 diabetes have to do with Parkinson's? It turns out quite a lot. It's quite well known and well recognized that if you have type 2 diabetes, you have a very strong chance, an increased risk factor of developing Parkinson's later on in life. And when you look mechanistically, the two conditions share some very, very common pathologies, such as impaired insulin signaling, mitochondrial dysfunction, and increased inflammation. So over the last decade, a lot of research has gone on to try to link the two. And a team at UCL in London published a paper back in 2013 showing the results of a clinical trial they ran on 40 patients, Parkinson's patients, where they treated with exenatide over 12 months. And what they found was that those patients treated with exenatide exhibited much better motor symptom scores and cognitive scores after the 12 months. And then when followed 12 months later, they actually showed that this effect persisted another 12 months off treatment. They then repeated the study on 60 patients and published in The Lancet in 2017, causing some very, very excited media hype and obviously excitement around the potential for exenatide as a treatment for Parkinson's. There is, however, a downside to this story. Exenatide, liraglutide, and all these other GLP-1 analogs are set to lose patent exclusivity in the next couple of years. So their prices will go down to generic prices, about $1 a day. And while this sounds pretty good, it is good if you're a diabetes patient. For Parkinson's, however, this makes it commercially unattractive for pharmaceutical companies to develop these drugs and spend the $500 million in clinical studies to what will amount to a generic drug at the end of the process. This is where we come in. We knew that we were big believers in the science. However, for us to get this science into patients, we had to make sure that we had something inventive, differentiated, which would make it commercially viable, and then of course it had to be efficacious. So we started with the GLP-1 receptor, and we knew by and large, if you hit this GLP-1 receptor with an analog, it's neuroprotective. GLP-1 has a sister hormone called GIP, and if you hit that with a GIP analog, that's also becomes neuroprotective. So we went one more step further, and we asked ourselves, what if we combine the two? And then added what's called a CPP, or cellular penetrating peptide, to this molecule. Well, we looked back and said, yes, this is indeed inventive, differentiated, and hence commercially viable. Now all we needed to see, does it work? And I'm happy to report, yes, it works really well. Better so than the mono-acting GLP-1 analogs. This is the MPT mouse model of Parkinson's disease. I'm not going to get into details, but basically these mice express and exhibit Parkinsonian-like symptoms. They have less dopamine, they have motor symptom problems, they have balance problems, and they have weaker strength, basically. When we treat them with our drugs, what we show is we're able to normalize dopamine levels, we're able to improve their strength, their balance, coordination. And then when we did a head-to-head study versus exenatide, the only molecule with clinical data, we were able to show that we were superior at equal doses and equally efficacious at 1/10 of the dose. So this gave us a lot of excitement on our chances. This is where we are today. This is our development plan. We're right here, right at the beginning, about to start our regulatory tox studies. We've been very, very fortunate to have received the Creation House 10 million crowns and been seated at the BII. I sit there in a small little corner, and they've given us just enough to get us to phase 1. Over the next 12 months, we'll be going and raising additional capital. This is a big, long process, but we're very, 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 very happy to have been seated there. This is the team behind Korea Pharmaceuticals. We are comprised of pharmaceutical industry veterans and drug developers. In the middle is Professor Christian Holscher, really the world's expert in incretins in the CNS. To the right, Michael Thompson, actually the first person to have ever dosed GLP-1s in humans at his time at Novo Nordisk. And then myself, a veteran at Takeda Pharmaceuticals for the last 10 years, where I was working in our CNS strategy and portfolio, and also a global program lead, heavily, heavily inspired by Christian's work. Happy to introduce you to the rest of the team at the break, but suffice it to say that we are all super excited about our chances, about our lead compound KP405, and fully believe that we are going to transform the treatment paradigm in Parkinson's disease, ultimately improving and extending the lives of millions. Thank you.
Thank you very much, Ian. I hope the analogy made sense now. I think it was pretty good. The next presenter is Peter. Peter is from the company Improter. They were also a part of the last batch at BA. You came in as a crazy scientist. Now you have built an amazing team in BI and is known in the office as the Björn Borg of vaccines. So take it away.
Well, thank you. Yes, so Improter, we are an immunotherapy company, and that means that we are trying to trick the immune system into recognizing cancers and kill them. So in our case, we do this by targeting viruses that are hidden within our genome and constitute a missed class of opportunity for treatment of cancer. So the team here is based upon research that I did as a group leader at the University of Copenhagen, but now I have spun out. We're an independent biotech company, so that means that actually, in this case here, it means that we are situated with access to labs at BI, and we have our own IP. We control and own the core IP for our technology. We have 9 employees now. They should be considered as coming up from 1 employee last year. And lastly, because of Novo Nordisk BI, that gave us the 10 million convertible note in January. Using this, we could then attract further funding from the Danish Innovation Fund in March and April. Now we've attracted 15 million. So thank you for this good start. I'll explain what we're doing. I will try to change slides. It works. Thank you. So the cancer immunotherapy space. Cancer immunotherapy is the most rapidly growing subdiscipline in oncology, probably maybe the largest indication in the medical space. This is the biggest obstacle. Cancer is the biggest obstacle for extending lifespan in human. So the market size is growing so rapidly because a new treatment has occurred, new principle that works and can cure cancers. Immunotherapies work, they cure them, but they only work if the immune system has already been activated. So a cancer that triggers the immune system, you can give new drugs, and they set the immune system free to kill the cancers. That provided a clear rationale for a number of new therapies that are trying to harness more immune system reactivity, raising cells that can kill more cancers, and then they can be treated by these new drugs. The problem is, this is the basis for about 2,000 clinical trials right now. So you might think this is a rather crowded space. But the problem is that they all depend on what we call here immunogenic antigens. What is that? That's mutations. So the immune system is normally tolerant to ourselves. So if you have your own genes, you should not react to them. Mutations can be seen as foreign by the immune system, and then we can induce them to react. That also means that all this fuss now, these 2,000 trials, are focused on something caused by smoking and sunlight, as a rule. The vast majority of cancers will have too few mutations to be targeted by either the current or the emerging treatments. We need something else that can persist in this space, that is shared and towards which we have no tolerance. We found that, and here we go. Yes. So what we're focusing on now is what is called endogenous retroviruses. So these are relics of past infections in our primate ancestors. They integrated into our germline, so became part of our DNA, and they multiplied. They're now 8% of our genome that consist of endogenous retrovirus DNA. Now, in there, they are silenced, and this is a big deal for our immune system, that we can silence retroviruses. Otherwise, we would be tolerant to them, and our next infection will kill us. So we have to be silent. They have to be silent, and we cannot be tolerant to endogenous retrovirus. That's incompatible with survival. Now, what turns out is now that they are required for cancer development. And what does that mean? That means that if you have cancer and take away their endogenous retrovirus genes, the cancers can no longer form. If you have cell lines that are immortalized so they can keep on proliferating, introducing these genes will now cause them to become cancers. Most spectacularly, if you can induce immunity to these genes, this is hard, I'll get to the point in a moment, but it can be done, then animals will become resistant to new cancers, even if they're formed by mutations. So you can give animals a carcinogen that will cause them to mutate and cause cancers. And there's no developmental path of cancer development that can bypass this requirement. So coupled with the lack of tolerance, this makes them an ideal target for immunotherapy. Now the next obvious question is, why haven't this been done before? And here's why. So the main target on the surface of cancer cells was also on the surface of virus particles in the past. This is the envelope protein that mediates the attachment of a virus to its cell target. Now when a virus does that, it puts in its DNA or its RNA into the cell, and the cell wants to kick back and defend itself. Viruses counteract this with an immunosuppressive domain, and that suppresses the immune system. In cancers, this becomes cancer-specific immune suppression. Now, this made these genes into quite poor vaccine genes, and results were poor in the past. We decided to go around this problem and solve it, and spend effort to it. So we basically introduced these genes in viruses that would by themselves cause this inflammation, viruses that didn't use this pathway. and didn't have this signal. So we could induce inflammation. We then did our final change, which is point mutation in this domain. And using a virus and these point mutations, now we can get robust immune responses, and this is basically animals measuring an immune response, cellular response in the spleen of a mouse. Now, the interesting thing is when you have that now, so now we've made immune responses to something that is immunosuppressive. There's no tolerance towards it. What happens then? Then we get curative cancer therapy. In this case here, you're seeing animals, they're getting a colorectal cancer at day 0. We give them the vaccine some days later, the cancers grow up, and they disappear. Now, this has been seen in some cases before. What you have never seen before is what we did next. We took the surviving animals, in this experiment here, more than 20 of them in different conditions, and then we gave them a new cancer, not the same one. They got a colorectal cancer first, we gave them a breast cancer, and now they're resistant to breast cancer, so they cannot get other cancers. That has never been shown before. This is really showing the potential of this technology. Right, so this is where we are from 10,000 feet. We raised the funding to get started. We're spending this funding on 9 people, of course, and on what you call the technical translation. And that is basically taking some technology and making it into a commercial product. We need to have something that can be produced as GMP and be injected into humans as an immunotherapy. That's the task. We work on a clinical development plan. This has taken time because there's many options in a technology like this. But we've been interacting with the KOLs locally and abroad, some of the key people in the field. We've selected on a patient group with a large medical need and a good chance of getting patients up. The clinicians are confirming this. And we are basically taking this plan now and showing it to the pharma partners that will need to take it forward after proof of concept studies. Where are we going to go in the next 12 months? We need to raise more money. We're guessing we need to raise 50 million euro to take this, our first vaccine candidate, into human phase 2 trials for efficacy studies. So this is the budget for one target. There's options to diversify on this, which will require more money. But this is what we're aiming at now. And this is the goal, to validate that this principle that is working so broadly and so remarkably effective in animals can also work in humans. Now the team, the core management team, you saw all the scientists in the pictures before. But this is Matthias Kroll. He came from a distinguished career in GSK, now in large biotech, small pharma, as you would call it, in Bavarian Nordic. So the expert, the biggest company in this field, we recruited him here. Nikolaj Høj Nielsen is a serial entrepreneur. He is responsible for our finances. He has been doing nothing but raising money for small startups in many, many years. We're saying we both have 20 years of experience. Anne-Marie Andersen, she's our CTO. She's also here in the audience. She developed the core technology that we are now building on and came back from Oxford for this project. And myself, well, I'm here to talk to you afterwards. Thank you.
Thank you so much, Peter.
Thank you.
For keeping our guests busy. I hope the results was something useful for you.
I forgot to say that Proxy Biotech received investments from all three of us, so we agree with you, audience.
Very good. Okay, thank you very much. We come to an end. I think we can all conclude. We saw fantastic pitches today. Three of our companies that are in our creation house, and the 4 teams graduating from the BAA class. Also, I think we clearly showed or seen what the BII team can do to any team that come in. So many salutes, I think, to the BI team, and particularly Monica and Christian, who has really been driving this program. So now I guess it's time to go to some place close on the corner here. It's called Bruce. You see, I come very often in Nørrebro. We are looking very much forward to welcome you all there for a small glass and celebrate this fantastic day. And then we look very much forward to see you all again in November sometime. Date is not fixed, but for our next demo day. Thank you very much.